1944, Oswald Avery and his colleague discovered the first evidence that gene is made of DNA, which is the carrier of the genetic information. In 1953, James Watson and Francis Crick discovered the structure of DNA, which is double helix. This is one of the most significant discoveries that opens the door for genetic engineering. An important step that leads to the advancement of genetic modification happens in 1973. Stanley Cohen and Herbert Boyer developed techniques to chemically cut and splice strands of DNA at specific places in the sequence. An enzyme called restriction enzyme cuts the code for a specific protein, while Cohen added a technique to introduce these DNA sequences into bacteria and yeast cells. This leads to the first recombinant DNA using bacteria gene. The first genetically engineered organism to be patented is oil-eating bacterium from Exxon Oil Company in 1980. 2 years later, humulin, which is a type of insulin produced by genetically engineered E. coli bacteria, appears on the market. However, the genetically engineered food is first sold in the grocery store shelf in 1994. It is flavored sour tomato which can resist rotting. An antisense gene is added in order to interfere with the production of the enzyme polygalacturonase, which normally degrades pectin in the cell walls and results in the softening of fruit. The product was approved by the U.S. Food and Drug Administration for sale. The data shows that in 2010, 93% of soybean grown in the USA is genetically modified, as well as 86% of corn and 90% of canola, and these GMO crops are then processed to be other products such as corn syrup, cornstarch, corn oil, canola oil, and soybean oil in mayonnaise, salad dressings, bread, and snack food.
For genetic modified crops or GM crops, there are mainly 4 steps. First, search for an organism with the desired trait and find the gene responsible for that trait. For example, let us consider Bob. We want him to have cute freckles and glowing antennae, so we need to look for an organism with those traits. Luckily, we found John with the following traits: good freckles and fluorescent antennae. Then we take his gene, the same species as John, but without freckles or fluorescent antennae, to compare their gene to find the gene that leads to the traits. This process is called comparative analysis. Next, isolate the gene of interest out of the organism and construct the structure of the gene. Now that we know the location of the destination gene, we need to take the gene out of John and construct them so they could work properly. Here we use restriction enzyme, which cuts the DNA at a particular region and isolate the desired gene from other undesired substances. After that, we multiply the isolated gene through a process called polymerase chain reaction, or PCR, and begin to prepare the structure. The structure to be prepared is called plasmid, which is a circular ring of DNA. Since with only the gene itself, the nucleus of the target does not know what to do with it, so we create plasmids to write commands to tell the target what to do with the gene. Our plasmid ring will have a region called promoter, which tells the RNA polymerase where to start. RNA polymerase acts like a reader machine for transcription DNA. Next is the gene of interest. Then we have reporter region, which expresses some noticeable expression so that we can identify which cell has successfully received the transgene. Then we need terminator region, which terminates the transcription of RNA polymerase, or basically telling it to stop.
Third, choose a method for inserting the prepared genome. There are 2 methods which are majorly used: agrobacterium and GeneGun. For the first method, we use the ability of agrobacterium, which can inject its plasmid into plant causing tumor in plant. By using this, we can replace the tumor-inducing plasmid with our plasmid and use it like a Trojan horse to deliver our plasmid into the target's nucleus. However, the downside of this method is that it has low yield, so let us introduce the second method, gene gun or biolistic. Choose DNA-coated metal particle into the target seeds, hoping that some bullets will get into the nucleus and do its job. However, despite having higher yield, more than one set of DNA may end up in one nucleus, or the cell structure might be damaged by the bombardment. So the researchers will have to choose which technique to use depending on circumstances. Last, screen the product and grow them in controlled environment. With the reporter region mentioned earlier, we can screen seeds which have successfully received the desired trait.
There's a frequently asked question what makes genetic modification different from traditional crossbreeding. In traditional crossbreeding, two plants with desired trait are crossed together to get a new plant with both of the traits from its parents. But this process may take a lot of time in order to find the successful offspring. Moreover, the yield of crossbreeding is much lower. The second difference is that sometimes the desired trait is taken from organisms that is impossible for them to breed, like tomato and bacteria. Thus, genetic modification has expanded the limit of what trait we can have from crossbreeding.
How do you think about GMO, genetically modified organisms?
I feel insecure to GMO food. If I eat it a lot, then I think it will affect my gene too.
The GMO system has been existing for more than 20 years. Due to the latest research, there is no one case that people's genes change because of consuming too much GMO products. Also, for the livestock which are fed by GMO crops, there is no evidence proves that their genes have been modified.
The GMO food might cause cancer or allergies, so personally, I don't want to take the risk to eat it.
Before modifying the gene of the organisms, we precisely control the gene fragment that we are going to insert and make it as a pattern. As long as every GMO product is double-checked by the government before they sell it, there will be no problem since we have controlled the gene fragment and have well studied of that.
Isn't GMO will be harmful to environment? Like the population of bee are decreasing dramatically. We shouldn't plant any GMO product, and the bee also because like pollinator insect will be harmed and extincted.
Calm down. This result shows that the current bee population is the highest in the past 20 years, which perfectly proved the fact that GMO products didn't affect the bee population. Also, the population of insects is influenced by more than one factor. Climate changing, plant fires are more critical to their survival.
I think it's impossible to say no to all GMO foods. And I've heard that the bananas in the supermarket now are all genetically modified. And the old bananas, they had huge seeds and which could not be swallowed easily. But since I ate so much so far and remain healthy, I think I will continue consuming GMO foods.
Yes, you are definitely right. GMO products have been widely spread throughout the world and frequently appear in our daily lives. For example, soybean, papaya, corn. There are so many things that we have already eaten in our life, I bet. So try not to be so afraid to GMO products and embrace them.
I feel like that GMO foods increase the price of the products.
After the modification of gene, GMOs are more easily to be planted and are more resistant to the harsh environment. Therefore, they can produce more products in the shorter time with the help of better gene, and it decreases the cost too, thanks to GMO. Last year, in 2017, the first genetically modified animal was commercialized. Salmon, after 25 years of research and development, entered Canadian market. It takes 18 months, 2 times faster than the normal salmon to breed and grow to its full length. Many other animals such as disease-resistant pigs and chickens are still under research. Though we should not expect much in these next few years, it takes a long time and process to get to the desired result. According to ISAAA, in 2016, 185 million hectares of land were planted with biotech crops, and the vast majority consisted of soybean, maize, cotton, and canola. Almost all of this area, over 99%, contain crops resistant to herbicides, insects, or both. What we can expect in the next few years is perhaps a solution to the current big challenge. GM crops tolerant to glyphosate have caused over-reliance on this single herbicide, causing an ever-increasing number of weed species to evolve resistance to it. Pests are also growing resistance to insect-resistant crops, making the crops once again vulnerable for attack. Other than that, disease-resistant banana, wheat, and potatoes, along with drought-tolerant sugarcane and maize, are all currently under research in institutions and laboratories. These researches focus more on aiding the farmers than increasing yield. Yet, as we saw from the interview, many people have a false negative opinion about GMO food. Even after a successful research project, it will take another few years for it to be accepted and commercialized. Another notable research is now being done by the Bill and Melinda Gates Foundation that aims to create cereal crops that can fix their own nitrogen. If successful, it will be a game changer for the whole farm industry, where a cost of using and producing fertilizer will be dramatically reduced. It isn't just the uses of GM technologies that are changing, but the technologies themselves. Many scientists are turning towards genome editing tools, the most famous of which is CRISPR-Cas9. These powerful tools can be used to introduce new genes into a plant or an animal, or to make more subtle changes in individual DNA-based pairs. They are often much faster and cheaper than older techniques of genetic modification, potentially allowing smaller players onto the field. Genome editing is not only a technological triumph, but an important distinction in the regulatory system. Countries such as the United States and Argentina have deemed that genome-edited plants must be regulated in the same ways as GMOs, which opens up the possibility of bringing new crops to market far more quickly and cheaply. Okay everyone, that's it for today. I hope you have understood about the history and the technology that go behind GMO food. As you have learned, there are several misconceptions going on about the GMO food. We hope today's video was able to clarify the facts about them. Please share what you have learned with your family and friends and have a nice day. Bye bye.